We've been exploring human adaptations and how we have evolved into such a successful species. So far, we've focused on these adaptations and why they've been so advantageous. But where did these adaptations actually come from? The answer lies in our DNA. Every living thing on Earth, at least that we know of so far, is built from a blueprint made of deoxyribonucleic acid, otherwise known as DNA. DNA is a complex molecule composed of a sugar phosphate backbone and 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. It forms a double-stranded helix where specific base pairs always bond together: adenine with thymine and cytosine with guanine. While the structure of DNA is the same across all species, what makes everything different is how these bases are arranged or sequenced. But if life started from simple bacteria that merely copy themselves, why isn't all DNA identical? What causes new DNA sequences and therefore new traits and adaptations to arise? Let's start by looking at how DNA becomes a trait. Here's the thing: DNA doesn't actually do the building itself. In fact, it never even leaves the nucleus. It's like a sacred library in the cell, keeping those precious instructions safe and sound. So how do these instructions get communicated to the rest of the cell? Enter an enzyme called RNA polymerase. This enzyme reads the DNA code and makes a copy that can leave the nucleus. This process is called transcription. Once outside the nucleus, this copy, called messenger RNA, or mRNA, gets read by a ribosome. Ribosomes are tiny structures in the cell that translate the mRNA's instructions into proteins. They do this by reading the mRNA in groups of 3 bases called codons. Each codon corresponds to a specific amino acid, which the ribosome adds to the growing chain to build a protein. Lucky for us, codons are also redundant, where multiple codons can refer to the same amino acid. This process continues until the ribosome reaches a stop codon. This step is called translation. For example, one protein might be melanin, which accumulates to give pigmentation to hair, skin, and eyes. So this covers how DNA is responsible for characteristics, but it still doesn't explain the variation we see between and within species. How does DNA change, shift, or even grow over time? Mutations are changes in the DNA sequence that can occur for a variety of reasons. Some mutations happen spontaneously during DNA replication when the cell makes a mistake while copying its genetic material. Others are caused by external factors, such as exposure to UV radiation, chemicals, or even viruses. These changes in the sequence can be small, like a single base pair being swapped, inserted, or deleted. Or they can be larger, affecting entire sections of the DNA or even entire chromosomes. Because of transcription and translation, mutations can have a range of effects. Some may be completely harmless. Remember that bases are read in groups of 3 called codons that correspond to an amino acid. Sometimes the mutation doesn't change which amino acid is being used because those codons are redundant, and so the mutation ends up being harmless. Others can be harmful. If a codon is altered enough, it can correspond to the wrong amino acid, causing diseases or malfunctions in proteins. Even more severe, it can cause an early stop codon that would prevent the protein from being made. But on rare occasions, mutations can be beneficial. If a mutation gives an organism an advantage in its environment, such as a better camouflage or resistance to a disease, that mutation may be passed on to future generations, driving evolution. Over time, beneficial mutations accumulate, helping species adapt to changing environments. One fascinating example of a beneficial mutation is lactose tolerance. For most mammals, the ability to digest lactose, the sugar found in milk, fades after infancy. This is because the enzyme responsible for breaking down lactose, called lactase, stops being produced. However, in some human populations, a mutation in the DNA that controls lactase production allows this enzyme to be made throughout adulthood. Lactose tolerance is a great example of how mutation can cause a trait that would be advantageous given the environmental hazards a population could be facing. Over generations, this led to an increased frequency of the mutation in certain regions, such as parts of Europe and North Africa, where food scarcity could become a problem during certain months of the year. In contrast, populations that historically didn't rely on dairy farming are more likely to have lactose intolerance, where lactase production decreased after childhood. This is a clear case of natural selection acting on a genetic mutation, shaping human evolution in response to diet and lifestyle. While mutations can lead to novel traits that are adaptive, it's important to remember that a majority are either neutral or harmful. It's unlikely that humans will ever acquire superpower mutations like X-Men, at least not for many thousands of years, and yet a series of unlikely, beneficial mutations have given our species undeniable advantages that allow us to survive in virtually any climate, solve any problem, and dominate any food chain.